Welcome, deep divers. Have you ever paused to think about your cells, those tiny, tiny powerhouses, and wondered how they keep everything perfectly organized? I mean, how do the cells, busy workers, the proteins, always know exactly where to go? It's a level of internal logic and precision that's truly astounding.
That's the fascinating puzzle we're diving into today. The intricate world of intracellular organization and protein sorting. We're drawing our insights from molecular biology of the cell, 7th ed, really trying to unlock how cells create and maintain their incredibly complex internal structure with such, well, efficiency.
Right. And our mission here is to pull out the most important nuggets of knowledge from this incredibly dense material, giving you a real shortcut to being well informed about the cell's internal blueprint. Expect some surprising facts and plenty of those aha moments. We'll explore everything from the way cells separate different molecules in space to the distinct membrane-enclosed compartments, as organelles, and even these dynamic self-assembling things called biomolecular condensates.
Yeah, those condensates act like specialized biochemical factories or maybe temporary storage depots. Super interesting.
So let's begin at the beginning, the sheer scale of the challenge. An animal cell can contain about 10 billion protein molecules, maybe 10,000 different kinds. And here's the kicker. Almost all of them start their journey in the cytosol, that general sort of jelly-like space that fills the cell outside the organelles. So the fundamental question is, how does the cell ensure that all these proteins end up in exactly the right place?
It's a fundamental biological problem, isn't it? Eukaryotic cells solve it through this brilliant strategy we call compartmentalization. Think of membranes not just as simple boundaries, but as, well, clever architects. They vastly increase the surface area available for vital reactions like making lipids or generating energy, and crucially, they create functionally specialized spaces. This means diverse biochemical reactions, each needing maybe different conditions like pH or specific enzymes, can all happen simultaneously without messing each other up.
And when we talk about these compartments, we're dealing with some very familiar names, right? You've got the nucleus, the cell's control center, housing the genome, and where DNA and RNA are made. Then there's the cytosol, which is actually over half the cell's volume, handling a lot of the general protein synthesis, degradation, and metabolism. And of course, the endoplasmic reticulum, or ER, which is just astonishing. It makes up about half the cell's total membrane area.
Yeah, the ER is huge. It's
Okay.
responsible for building proteins and lipids. It also acts as a vital calcium store. And from the ER, products often move to the Golgi apparatus, which acts like the cell's post office, receiving, modifying, dispatching molecules. Then we have lysosomes, essentially the cell's recycling centers, packed with digestive enzymes to break down old organelles and large molecules.
Right, the cleanup crew.
Exactly. And endosomes are like pre-sorting stations on the way to the lysosomes. And of course, the energy powerhouses, mitochondria, and in plant cells, chloroplasts.
Yeah.
Lastly, peroxisomes, small but mighty compartments dedicated to oxidative reactions.
It's not just what they are, but how much of them there is that's mind-boggling. You said these membrane-enclosed compartments together occupy nearly half the cell's total volume.
Pretty much, yeah. And the ER. In liver cells, its total membrane surface area can be up to 25 times that of the plasma membrane. It's truly the cell's biggest internal organ, in a way.
Oh.
And that abundance and specialization aren't arbitrary. They're perfectly tuned to the cell type's specific functions. Take plasma cells, for example. They produce and secrete a huge amount of antibodies, so they're absolutely crammed with rough ER. Muscle cells, needing rapid calcium release for contraction, have a highly specialized smooth ER called sarcoplasmic reticulum alongside, an army of mitochondria for energy. Each cell fine-tunes its internal layout.
Okay, here's where it gets really interesting for me. How did all this amazing complexity evolve? Because this intricate system didn't just appear out of nowhere, did it?
That's a crucial evolutionary question. We have two main hypotheses for how these organelles came to be. First, the plasma membrane invagination hypothesis. Imagine an ancestral prokaryotic cell, much simpler. It started forming protrusions or folds in its outer membrane, probably to increase surface area. Over time, these folds deepened and eventually pinched off, forming internal membranes.
Ah, like little pockets folding inwards.
Exactly. And this elegantly explains why structures like the ER, Golgi, endosomes, lysosomes, peroxisomes, and even the nuclear envelope are all topologically connected or can communicate via these little transport vesicles. They're all sort of related to the outside world, topologically speaking.
Yeah.
The nucleus itself, with its distinct environment for genetic material, is thought to have evolved from the ancestral plasma membrane wrapping around the cell's genome.
Okay, that's one theory. What's the other?
The second hypothesis is endosymbiosis. This explains the origin of mitochondria and chloroplasts. The idea is that ancient symbiotic bacteria were engulfed by ancestral cells and then, over millions of years, evolved to become these distinct organelles. This is why they uniquely have double membranes and are essentially isolated from the vast vesicular traffic connecting the other organelles. It's also why their own protein transport machinery shares striking similarities with modern-day bacteria. A real biological echo.
With cell within a cell, almost. Okay, so membranes are one way to organize, but what about other structures that aren't membrane-bound? Cells have organization beyond just those internal sacs, right?
Absolutely. This brings us to biomolecular condensates. Think of them as tiny, self-assembling molecular clubs within the cell. Instead of a membrane wall, they form through a process called liquid phase separation, much like oil droplets forming in water, but at a microscopic molecular level.
Liquid phase separation sounds fluid.
Yeah. It is. This allows specific groups of proteins and RNA to gather and perform reactions together without being contained by a traditional membrane. They form through weak but numerous interactions between specific scaffold molecules, which then recruit client molecules, effectively concentrating them in one place.
And these aren't just theoretical, we see them in action. The largest and most famous is the nucleolus, right? The cell's ribosome assembly factory.
That's the one. Others include peronoids, vital for carbon fixation in algae, and stress granules, which temporarily store components related to translation when a cell is, say, under stress, like heat shock. What's truly fascinating is their dynamic liquid-like properties. They're typically spherical, can deform, and even fuse or split, almost like tiny living droplets.
But they can solidify sometimes, too.
Yeah, if the interactions within them become stronger, they can transition into more gel-like or even solid states, like the structured concentric layers you see in the nucleolus.
So they function as biochemical factories.
Exactly. By concentrating specific enzymes, they significantly accelerate multi-step reactions. The pyronoid, for instance, concentrates key carbon-fixing enzymes, boosting efficiency even in low CO2 environments. This precise organization helps prevent unwanted side reactions and maximizes efficiency. And it's not just understanding them. Scientists are now even learning to engineer artificial condensates for specific biochemical tasks. Pretty cool stuff.
And they're not static, which must be crucial for a cell's ability to adapt quickly.
Totally crucial. These condensates dynamically assemble and disassemble. This is often controlled by subtle chemical modifications to proteins, like adding or removing a phosphate group phosphorylation, or by changes in cellular conditions like temperature or pH. This allows for incredibly rapid adaptation, like the swift formation and dissolution of stress granules when a cell encounters or recovers from, well, stress. It's like a cellular pop-up shop.
Okay, so we have membranes, we have condensates. How do proteins actually move between all these different places?
Right, navigation. There are basically 4 fundamental ways proteins move between compartments. 1, protein translocation. This is direct transport across a membrane, like a protein threading its way from the cytosol into the ER or a mitochondrion. The protein usually has to unfold to snake through a special protein channel.
Okay, direct crossing, what else?
2, gated transport. This is how molecules move between the cytosol and the nucleus. It happens through these selective gates called nuclear pore complexes. Small stuff can sneak through, but larger molecules need, like, a special pass.
Got it. Gates for the nucleus.
3, vesicular transport. This is where tiny membrane-enclosed bubbles or vesicles ferry proteins between compartments that are topologically equivalent. Think of the journey from the ER to the Golgi. The proteins never actually cross a membrane during this process. They stay inside the vesicle or stuck in its membrane.
Like cargo in a shipping container.
Precisely. And 4, engulfment. This involves double membrane sheets wrapping around parts of the cytoplasm. You see this in autophagy, where the cell recycles its own bits, or during the reformation of the nuclear envelope after cell division.
And for entering those biomolecular condensates we just talked about, how does that work?
That's simpler. It's usually direct physical binding, no membrane crossing, just a specific interaction, almost like having the right kind of Velcro patch, like a direct sorting signal.
So these sorting signals are key, like the cell's internal postal codes.
Exactly. They're typically specific sequences of amino acids within a protein, or sometimes a unique 3D arrangement called a signal patch. These signals, like a short sequence at the N-terminus for the ER, or maybe just 3 amino acids at the C-terminus for peroxisomes, are recognized by specific sorting receptors. Scientists have done clever experiments. If you take an ER signal and attach it to a protein that normally stays in cytosol, boom, that protein suddenly goes straight to the ER.
So the signal is both necessary and sufficient.
Exactly. It proves the signal dictates the destination. And often, these signals rely on simple physical properties like hydrophobicity, how much they dislike water, rather than a super precise amino acid sequence.
This brings up an interesting point about how organelles themselves are built. You mentioned the ER, mitochondria, and chloroplasts need pre-existing protein translocators to grow, so they can't just be made completely from scratch.
That's right. They must be inherited, passed down from the parent cell when it divides. You need a template, essentially. But fascinatingly, other organelles like lysosomes and those biomolecular condensates we discussed, such as the nucleolus, can be constructed entirely de novo, from scratch. They self-assemble from their individual components. Their bits and pieces can be randomly distributed during cell division, and then they'll spontaneously reassemble into functional structures.
So it's a mix of inheritance and self-assembly. Clever.
Very clever. Each strategy is optimized for the specific organelle.
Okay, let's zoom in on a true superstar organelle, the endoplasmic reticulum. So it's over half the cell's total membrane.
Yeah, it forms this incredible net-like labyrinth of branching tubules and flattened sacs. It's even continuous with the outer nuclear membrane, which really highlights its central role in making lipids and proteins, plus acting as that vital calcium storage vault.
And it comes in different flavors, right? Rough and smooth.
Exactly. The rough ER is studded with ribosomes, giving it that bumpy appearance. This is the primary site for making proteins destined for secretion or for insertion into membranes, or delivery to other organelles like the Golgi and lysosomes. Cells that secrete a lot of protein, like those pancreatic cells making digestive enzymes or plasma cells pumping out antibodies, are absolutely packed with rough ER. Makes sense, right?
Totally. And the smooth ER.
The smooth ER lacks ribosomes. Looks smooth. It has a more varied set of functions. All cells have some transitional ER, which is where transport vesicles bud off on their way to the Golgi. But in specialized cells, the smooth ER really expands. For instance, cells making steroid hormones have lots of it. Liver cells use their smooth ER for detoxifying drugs, using enzymes like cytochrome P450 and for making lipoproteins. And a crucial universal function is calcium sequestration, storing and releasing calcium ions. Absolutely vital for things like muscle contraction, where it's called the sarcoplasmic reticulum.
And you mentioned it makes close contacts with other organelles, like a cellular handshake.
Yeah, these aren't just random bumps. They're very specific membrane contact sites, crucial for direct communication, and really importantly for lipid transfer between the ER and things like mitochondria or the plasma membrane.
Okay, let's unpack this. How do we even study something so intricately woven into the cell? It sounds like a nightmare to isolate.
Yeah, it was a challenge. Scientists came up with a clever trick. They can gently break open cells, which causes the ER to fragment and reseal into tiny closed vesicles called microsomes. These microsomes are like miniature, authentic versions of the ER, still capable of doing their jobs. And you can even separate the rough ones with ribosomes from the smooth ones using centrifugation based on density. This has been invaluable for figuring out exactly what the ER does.
Here's where it gets really, really interesting for me. The discovery of how proteins actually get into the ER, that signal hypothesis must have been huge.
Oh, it was revolutionary. This is the signal hypothesis.
Huh.
Scientists noticed that proteins destined for secretion were initially slightly larger when made in a test tube without ER bits. But if ER microsomes were present, the protein ended up the correct size and, crucially, inside the microsomes.
So an extra bit gets snipped off.
Exactly. This led to the idea that these proteins have an extra signal sequence, like that postal code usually at their N-terminus, the beginning of the protein chain. This signal directs them to the ER membrane, and once it's done its job, it's cleaved off by an enzyme called signal peptidase.
So how does that signal sequence actually work? What recognizes it and guides it?
That's where the signal recognition particle, or SRP, and its partner, the SRP receptor, come in. The SRP is this really cool molecular machine. It has a hydrophobic pocket that grabs onto many different kinds of hydrophobic ER signal sequences as they emerge from the ribosome. And when SRP binds, it actually pauses translation, temporarily stops the ribosome from making more protein.
Why pause?
It's critical. It gives the ribosome enough time to be guided to the ER membrane by the SRP, where it docks onto the SRP receptor. This ensures the protein isn't just released into the cytosol where it doesn't belong.
Okay, so SRP brings the ribosome to the ER membrane. Then what?
Then the SRP hands off the ribosome to a protein translocator, a channel called the Sex61 complex. This complex forms a water-filled tunnel right through the membrane. When the protein signal sequence inserts into a specific gate in Sec61, it opens the channel wider and moves a temporary plug out of the way. This creates a continuous path for the polypeptide chain to thread directly from the ribosome tunnel into the ER lumen, the space inside the ER.
And this is happening while the protein is still being made?
Yes, that's co-translational transfer. The ribosome is literally pushing the growing protein chain through the channel, and this tight seal also prevents ions from leaking across the membrane. Very efficient.
But you mentioned earlier that import doesn't always happen co-translationally. What about post-translational import?
Good point. Post-translational translocation is actually more common in simpler cells like yeast and bacteria, but it happens in our cells too. Here, the protein is fully synthesized in the cytosol first. Then, specialized cytosolic chaperones like HSP70 proteins act like molecular escorts, keeping the precursor protein unfolded so it doesn't clump up before it reaches the ER. Once at the ER, it engages the Sec61 translocator, and accessory proteins actively pull or push the polypeptide into the ER lumen. One key player inside the ER is a chaperone called BiP. It binds to the emerging protein segments and uses ATP hydrolysis like a molecular ratchet, preventing backsliding and pulling the protein in.
So, if a protein is actually meant to live in the membrane, not just pass through, how does it get embedded there?
Great question. Transmembrane proteins use the very same SRP receptor Sec61 system. Their hydrophobic transmembrane segments act kind of like internal signal sequences. They enter the Sec61 channel, but then they slide out sideways through Sec61's lateral gate right into the lipid bilayer.
So they just step out of the channel into the membrane?
Pretty much. For single-pass proteins, their orientation, which end faces the cytosol and which faces the lumen, is determined by subtle features like flanking charges or the length of the hydrophobic segment. Some might use a standard N-terminal signal sequence to start threading, and then a later hydrophobic segment acts as a stop transfer signal, halting translocation and anchoring the protein.
And what about proteins that span the membrane multiple times?
Multipass proteins are more complex. They're woven into the membrane by successive hydrophobic segments that alternately act as start transfer and stop transfer signals, engaging the lateral gate in a precise back and forth pattern. It's like stitching the protein into the membrane fabric. They're also tail-anchored proteins with a single transmembrane segment right at their C-terminus. They get inserted post-translationally by a completely different specialized pathway involving a factor called GET3. And then there are GPI-anchored proteins. They start as transmembrane proteins, but then they're cleaved, and the luminal part gets covalently attached to a preformed glycolipid anchor called the GPI anchor in the membrane.
Wow, lots of ways to get stuck in the membrane. Okay, so once a protein gets into the ER lumen, it's like a rough draft that needs editing and folding, right?
Exactly. Proteins enter as long, unfolded chains. The ER lumen is packed with resident chaperones like BP, which we mentioned for post-translational import. BP is an HSP70 family member. It binds to exposed hydrophobic patches on unfolded proteins, preventing aggregation and using ATP hydrolysis to help them fold correctly. It's a cycle of binding and releasing, giving the protein chances to find its proper shape. And these resident proteins, like BP, have a special ER retention signal, usually KDEL at the C-terminus, that keeps them from accidentally leaving the ER.
And disulfide bonds are important too, right? Especially for secreted proteins.
Absolutely. Disulfide bonds linking cysteine residues add stability. The ER lumen is an oxidizing environment, which favors their formation. An enzyme called protein disulfide isomerase, or PDI, catalyzes the formation and, crucially, the rearrangement of these bonds. If the wrong bonds form initially, PDI can break them and allow correct pairs to form. It's like a molecular proofreader for bonds.
And then there's glycosylation, adding sugars.
Yes, a major ER function is N-linked glycosylation. A large, preformed 14-sugar oligosaccharide is transferred en bloc from a lipid carrier called dolichol onto specific asparagine residues in the sequence as an exterther on the growing polypeptide chain. This happens almost immediately as the protein enters the lumen, catalyzed by an enzyme complex called oligosaccharyl transferase, which is associated with the Sec61 translocator.
And these sugars aren't just decoration, are they? You mentioned they act as tags for folding status.
Exactly. That's a key quality control mechanism. Shortly after the sugar tree is attached, specific enzymes called glucosidases trim off 2 of the terminal glucose units. This leaves a specific structure that's recognized by ER chaperones, like calnexin, which is membrane-bound, and calreticulin, which is soluble in the lumen. These chaperones bind to the incompletely folded glycoprotein, holding it in the ER and giving it more time to fold.
So it's a cycle.
Yes. If the protein folds correctly, the last glucose is removed, and it's allowed to exit the ER. If it remains misfolded, another enzyme, glucosyltransferase, acts as a sensor. It recognizes misfolded proteins and re-adds a glucose unit, sending it back to calnexic and reticulum for another folding attempt. This cycle continues until the protein folds correctly or it gets targeted for degradation.
Right. Not every protein makes it. What happens to the ones that just can't fold correctly? The rejects.
The cell has a strict policy.
Yeah.
Get rid of them. This is ER-associated degradation, or ER. Misfolded proteins are recognized, often because they persistently expose hydrophobic regions or fail the glycosylation check. Then, surprisingly, they're transported backwards out of the ER through specific translocator complexes related to Sec61 but involving other factors back into the cytosol.
Retro-translocation. Back the way they came.
Essentially, yes. Once in the cytosol, they're immediately tagged with chains of ubiquitin by enzymes called E3 ubiquitin ligases. This ubiquitin tag is the signal for destruction. Then, large protein-destroying machines called proteasomes recognize the tagged protein and chew it up. AAA AT passes provide the energy to physically pull these stubborn, misfolded proteins out of the ER membrane or lumen. The trimming of other sugars, mannoses, on the end-linked oligosaccharide can also act as a timer, marking proteins that have spent too long trying to fold.
So the cell has a whole alarm system for when the ER gets overwhelmed with misfolded proteins, like if EROD can't keep up.
It absolutely does. This is the unfolded protein response, or UPR. It's a crucial stress response. When misfolded proteins accumulate in the ER, they bind to sensor proteins embedded in the ER membrane, activating them. There are three main parallel UPR pathways, each triggering signaling cascades that ultimately lead to changes in gene expression designed to alleviate the stress.
How do they work?
Okay, pathway 1, IRE1. When activated by misfolded proteins, IRE1 acts as an enzyme that splices a specific mRNA in the cytosol. This spliced mRNA then codes for a transcription factor that goes to the nucleus and turns on genes for chaperones and other ER quality control components. Pathway 2, PEER. Activated PEER is a kinase. It phosphorylates a key protein involved in initiating translation, which reduces overall protein synthesis globally, thus lessening the load on the ER. However, it paradoxically increases the translation of a specific mRNA, encoding another transcription factor that also boosts chaperone production. Pathway 3, ATF6. This sensor protein, when misfolded proteins accumulate, actually travels to the Golgi apparatus. There, it gets cleaved by proteases, releasing a fragment that goes to the nucleus and acts as yet another transcription regulator for UPR genes.
So multiple ways to signal help, need more folding capacity.
Exactly. The UPR tries to restore balance by increasing ER size and folding capacity. This is vital for cells that secrete lots of protein, like insulin-producing beta cells or antibody-secreting plasma cells. They rely heavily on robust UPR. But if the stress is too severe or prolonged and homeostasis can't be restored, the UPR pathways can switch gears and trigger apoptosis-programmed cell death. It's a way to eliminate a dangerously malfunctioning cell.
The ER isn't just about proteins, though. It's also a major lipid factory.
Indeed. The ER membrane is the primary site for synthesizing nearly all the cell's major lipids, including phospholipids, the main building blocks of membranes, and cholesterol. What's interesting is that phospholipid synthesis happens exclusively on the cytosolic leaflet of the ER membrane. Enzymes involved have their active sites facing the cytosol. To ensure the membrane grows evenly on both sides, a special protein called a scramblase non-selectively flips newly made phospholipids from the cytosolic leaflet to the luminal leaflet, equilibrating them across the bilayer.
So the ER membrane grows symmetrically, but other membranes aren't always symmetric, right?
Correct. Membranes, like the plasma membrane, have a distinct asymmetric distribution of lipids, which is crucial for their function. That asymmetry is established later, mainly in the Golgi, by different enzymes called flipases that selectively move specific lipids. The ER also produces ceramide, a precursor for other important lipids, like sphingomyelin and glycosphingolipids, which are synthesized mainly in the Golgi. So ceramide needs to be exported from the ER.
Okay, if the ER is the central lipid factory, how do other organelles, especially those not connected by vesicles, get their lipids? Thinking about mitochondria and chloroplasts again.
That's where those membrane contact sites we mentioned earlier become critical. These are specialized regions where the ER membrane comes very close, just nanometers away, to other organelle membranes. At these sites, specific lipid transfer proteins act like little shuttles. They can bind a lipid molecule in one membrane, shield its hydrophobic part, diffuse across the narrow gap, and insert it into the other membrane. This non-vesicular transport is essential for getting lipids like phosphatidylserine or cholesterol from the ER to mitochondria, for example, which aren't part of the main vesicle trafficking routes. It's a direct, targeted delivery system.
Fascinating. Okay, moving on from the ER powerhouse. What about peroxisomes? They sound chemically active.
They definitely are. Peroxisomes are small membrane-bound organelles found in virtually all eukaryotic cells. They're major sites of oxygen utilization. They contain high concentrations of oxidative enzymes, like catalase and urate oxidase. They get their name because they generate hydrogen peroxide, H2O2, a reactive molecule, during various oxidative reactions. But they also contain tons of catalase, which efficiently breaks down H2O2, either using it to oxidize other substances or converting it directly to water and oxygen. So they both produce and neutralize H2O2.
What kind of reactions do they handle?
A key role is breaking down fatty acids through a process called beta oxidation. Although mitochondria also do this, peroxisomes handle very long-chain fatty acids. They're also important in detoxification, especially in liver and kidney cells, breaking down toxins like alcohol. And crucially, they synthesize certain essential phospholipids, including plasmalogens, which are abundant in the myelin sheath that insulates nerve axons.
So defects would cause nerve problems.
Exactly. Defects in plasmalogen synthesis due to peroxisome dysfunction are linked to severe neurological diseases. Plants have specialized peroxisomes too, involved in photorespiration in leaves and converting stored fats into sugars during seed germination in glycosysomes. And yeasts can adapt their peroxisome content depending on their food source. Very adaptable organelles.
How do proteins get into peroxisomes? Do they use the ER pathway?
Mostly no. While some peroxisomal membrane proteins might initially insert into the ER and then bud off in precursor vesicles, the vast majority of peroxisomal proteins, especially the enzymes inside, the matrix proteins, are imported directly from the cytosol after they've been fully synthesized. They use specific short sorting signals. The most common one is just a serine-lysine-leucine sequence, SKL, right at the C-terminus. This signal is recognized by a soluble receptor protein in the cytosol called PEX5. PEX5 ferries the cargo protein to the peroxisome membrane, where it interacts with the docking complex. Remarkably, the protein doesn't necessarily need to unfold to get imported. Even fully folded or oligomeric proteins can pass through the peroxisomal translocator, which is still not fully understood. ATP hydrolysis is needed, particularly for recycling the PEX5 receptor back to the cytosol.
And if that import fails,
that leads to devastating inherited conditions like Zellweger syndrome. In these diseases, the peroxisomal protein import is defective. The peroxisomes are essentially empty ghosts because they can't import their necessary enzymes. This results in severe abnormalities in the brain, liver, and kidneys, usually leading to death in early childhood. It really highlights how vital peroxisomes are.
Okay, let's turn to the big energy producers, mitochondria and chloroplasts, double membranes, ATP synthesis. We know they have their own DNA, but you said most proteins come from the cytosol. How does that import work?
Right. Despite having their own genetic system, the vast majority of the 1,500 different proteins in mitochondria, and even more in chloroplasts, are encoded by nuclear genes, synthesized on cytosolic ribosomes, and then imported. Both organelles grow by importing these proteins into their various subcompartments: outer membrane, inner membrane space, inner membrane, and the matrix, mitochondria or stroma, chloroplasts. Chloroplasts have the additional thylakoid system inside the stroma.
So similar challenge to peroxisomes importing folded or unfolded proteins.
For mitochondria and chloroplasts, proteins generally have to be kept unfolded for import. They typically have an N-terminal signal sequence, often forming an amphipathic alpha helix, one side hydrophobic, one side charged. This signal sequence directs the precursor protein to receptor proteins on the outer membrane translocator complex, the TOM complex in mitochondria.
TOM for translocase of the outer membrane.
Exactly. TOM handles the initial import of almost all nuclear-encoded mitochondrial proteins across the outer membrane into the inner membrane space. From there, different paths lead to the final destination. Some proteins, like beta-barrel proteins for the outer membrane, are handled by the SAM complex, sorting and assembly machinery. Proteins destined for the matrix or inner membrane engage with inner membrane translocators, TIM23 or TIM2, translocase of the inner membrane. TIM23 moves proteins into the matrix and helps insert some into the inner membrane. TIM-22 is dedicated to inserting multipass intermembrane proteins, like metabolite transporters. And then there's the OXA complex in the inner membrane, which inserts proteins synthesized inside the mitochondria on mitochondrial ribosomes, as well as some nuclear-encoded proteins arriving from the matrix.
And this import is post-translational, right, after the protein is made?
Yes, definitely post-translational for mitochondria. Cytosolic chaperones like HSP70 bind to the precursor protein, preventing it from folding prematurely and keeping it import competent. They use ATP hydrolysis to do this.
What powers the actual movement across the membranes?
It requires energy from multiple sources. First, ATP hydrolysis by those cytosolic HSP70s for release. Second, crucial energy comes from the membrane potential across the inner mitochondrial membrane. It's negative on the matrix side. This electrical gradient helps pull the positively charged signal sequence through the TIM channel. Third, another HSP70 chaperone inside the matrix, mitochondrial HSP70, binds to the incoming polypeptide and uses ATP hydrolysis to act as a ratchet, pulling the protein through TIM-23 and preventing backsliding. And fourth, for some proteins getting folded in the inner membrane space, like small cysteine-rich proteins, import is driven by disulfide bond formation, tapping into the redox potential maintained by the electron transport chain.
Wow, multiple energy inputs ensuring it gets there. And chloroplasts, similar story.
Similar principles, different machinery, and different energy sources. Chloroplast import is also post-translational, proteins kept unfolded. They also have N-terminal signal sequences called transit peptides, recognized by outer membrane receptors and translocators. TOC complex translocon at the outer envelope of chloroplasts. Intermembrane translocation uses the Teaging complex. However, unlike mitochondria, chloroplast import across the inner membrane doesn't depend on a membrane potential. It's driven solely by ATP and GTP hydrolysis on both sides of the envelope.
And for proteins going all the way into the thylakoids.
They typically have a second signal sequence, a thylakoid transfer domain, which is unmasked after the first chloroplast signal sequence is cleaved off in the stroma. This second signal targets the protein to one of several different protein translocators in the thylakoid membrane pathways homologous to bacterial protein export systems, SEC, TT, and even an OXA-like pathway, again highlighting that endosymbiotic origin.
Okay, last major destination. The nucleus. The genome's control center. Double membrane, continuous with the ER, but punctuated by these huge nuclear pore complexes, NPCs.
Exactly. The nuclear envelope, with its inner and outer membranes, defines the nuclear compartment. The inner membrane has unique proteins that bind to chromatin and the nuclear lamina, a protein meshwork underneath that gives the nucleus structural support. And those NPCs are the gatekeepers. Traffic through them is massive and bidirectional, things going in. All the nuclear proteins synthesized in the cytosol histones, DNA and RNA polymerases, transcription factors, ribosomal proteins, things going out, all types of RNA, processed mRNAs, tRNAs, rRNAs, snRNAs, and assembled ribosomal subunits.
How do these pores manage to be so big yet so selective? Seems like a contradiction.
It's an amazing structure. NPCs are enormous, about 30 times the mass of a ribosome, built from roughly 30 different proteins called nucleoporins, arranged with beautiful 8-fold rotational symmetry. The key to selectivity lies in the central channel. It's filled with intrinsically disordered regions of certain nucleophorins, rich in phenylalanine and glycine, FG repeats. These FG repeats form a sort of hydrophobic, gel-like meshwork.
A sticky filter.
Kind of. Small molecules, under about 40 kD, can diffuse relatively freely through this mesh. But larger molecules, like most proteins and ribosomal subunits, can't pass easily. They get stuck, essentially.
So if large proteins can't just diffuse in, how do they get through? They need a pass.
They need a transporter and the right ticket. The ticket is a nuclear localization signal, NLS, typically a short sequence rich in positively charged amino acids, lysine, arginine. The transporter is a nuclear import receptor, part of the karyophrin family. These receptors bind to the NLS on the cargo protein. Crucially, they also have binding sites for those FG repeats in the MPC channel. So the receptor-cargo complex doesn't just diffuse, it interacts transiently with successive FG repeats, effectively dissolving or hopping through the meshwork. It's a rapid, facilitated transport.
But what gives it directionality? Why do proteins accumulate in the nucleus?
That's the clever part. Directionality is imposed by the small GT-pace RAN. There's a steep concentration gradient. RAN is mostly kept in its GTP-bound state inside the nucleus, thanks to a RAN-GTF anchored to chromatin, and in its GDP-bound state outside in the cytosol, thanks to a RAN-GP.
So high range GTP inside, high range GDP outside. How does that drive import?
Like this. The import receptor picks up its NLS cargo in the cytosol, where RAN is GDP-bound. It moves through the MPC. Once inside the nucleus, it encounters high concentrations of RAN-GTP. RAN-GTP binds strongly to the import receptor. This binding causes a conformational change in the receptor, making it release its cargo protein inside the nucleus. The now empty receptor, bound to RAN-GTP, travels back out to the cytosol. There, RAN-GTP triggers RAN to hydrolyze its GTP to GDP. RAN-GTP dissociates from the receptor, leaving the receptor free to pick up another cargo. The cycle drives net accumulation of cargo in the nucleus.
Brilliant. And export works the same way, but in reverse.
Essentially, yes. Nuclear export signals, NES, on cargo proteins are recognized by nuclear export receptors. But here, the binding of the export receptor to its cargo is stabilized by RAN-GTP inside the nucleus. So the receptor-cargo RAN-GTP complex forms in the nucleus and moves out through the MPC. In the cytosol, RAN-GP hits, RAN hydrolyzes GTP to GDP, the complex falls apart, releasing the cargo and the receptor. Directionality again, driven by the RAN gradient.
What about mRNA export? Does that use RAN?
Mostly no. mRNA export is a key exception and mechanistically distinct. It doesn't rely on karyophorins or the RAN cycle directly. Instead, as mRNA is processed and spliced in the nucleus, it gets coated with specific RNA-binding proteins, including an exporter complex. This complex guides the mRNA through the MPC. On the cytosolic side, an RNA helicase uses ATP hydrolysis to remove the exporter proteins, effectively pulling the mRNA out and preventing it from going back in.
Ensures unidirectional export.
And cells can control this transport, right? Turn import or export on or off. Absolutely. Regulating nuclear transport is a major way cells control gene expression and respond to signals. Access to NLS or NES sequences can be controlled. For example, a signal might be masked by phosphorylation or by binding to an inhibitory protein in the cytosol. A stimulus might then cause dephosphorylation or release of the inhibitor, unmasking the signal and allowing nuclear import. A classic example is the transcription factor, NF-AT, in T cells. When the T cell is stimulated, calcium levels rise, activating a phosphatase that removes phosphates from NF-AT, exposing an NLS, allowing it to enter the nucleus and activate immune response genes.
Makes sense. One last thing about the nucleus, what happens during cell division? The whole envelope breaks down, right?
In higher eukaryotes, yes. During mitosis, the nuclear envelope dramatically disassembles. Enzymes called CD keys phosphorylate key proteins, including the nuclear lamins and some nucleoporins. This causes the lamina meshwork to depolymerize and the MPCs to break apart into subcomplexes. The nuclear membranes retract into the ER network. This allows the spindle microtubules access to the chromosomes to segregate them. Then, late in mitosis, the process reverses. Phosphatases remove the phosphates. Lamins and MPC components reassemble around the separated chromosomes. ER membranes wrap around the chromatin, fusing to form the sealed nuclear envelopes of the two daughter nuclei. RAN-GTP, concentrated around the chromosomes by the chromatin-bound RAN-GEF, plays a key role in guiding this reassembly process.
It's just incredible choreography.
It really is.
What an amazing journey we've taken, really getting into the cell's internal organization, from those tiny sorting signals to the complex machinery of transport and all that quality control. What's truly fascinating here is how every single part, whether it's a tiny protein or a massive organelle, has a precise address and a sophisticated delivery system. It's not just efficient. It's a marvel of molecular engineering.
Absolutely. And thinking about all this precision, this dynamic adaptability we've discussed, it raises an important question, doesn't it? Given the incredible control cells exert over this internal chaos, how they manage traffic, quality control, and respond constantly, what might be the next frontier in understanding how they orchestrate all this? How do they really manage the complexity and respond to the ever-changing demands of life? What haven't we seen yet?
A great question to ponder. What is the next level of understanding? Well, that's something for us all to think about. We really hope you've enjoyed being part of our deep dive family today, and that you feel maybe a little more in the know about the invisible, yet incredibly organized world inside you. Until next time, keep diving deep.